Gnana Deeksha, concept-based digital learning. I am N. Kavita, lecturer in biotechnology from Telangana Social Welfare Residential Degree College for Women, Mahendra Hills. Students, today we are going to learn a very interesting topic, karyotyping of human chromosomes. Karyotyping. Karyotyping is a test used to identify the abnormalities in the structure and number of chromosomes of a person. When I said it is a test, you might be feeling it is a simple routine blood test. Dear students, sometimes while collecting the sample, you may feel it is a normal blood test. But it is not just a simple blood test. It is a very important and sophisticated test usually performed during genetic counseling. Learning objectives. At the end of the lesson, we will be able to know about karyotyping and karyogram. We can understand the reason for performing a karyotyping. We also come to know how to perform karyotyping. From this knowledge, we can able to identify normal and abnormal karyograms. Along with the applications, we also going to learn reasons for abnormal karyotyping. But students, what is the need to study the abnormalities of chromosomes? The blueprint for the formation, growth and development of an organism is provided by the DNA, and this DNA is packed into chromosomes. Any changes that affects the structure and number of the chromosomes can cause problems in the functions of the body's system. Sometimes these changes may result in genetic disorders, cancers, developmental delays, etc. Not only this, as you know, chromosomes are vehicles of inheritance. They can pass these changes from parents to children. If a person suffering with a genetic disorder, he could pass that disease to his children. So, it is very crucial to know the abnormalities of chromosomes for healthy next generation. Then, next question rises in our mind. Is it necessary for everyone to get tested for karyotyping? No, it is not necessary for everyone to get tested for karyotyping. There are certain conditions where your doctor may recommend you for karyotyping. So, when it is recommended, if a pregnant woman wants to know the abnormalities in the fetus, she must go for karyotyping. These type of tests usually called as prenatal screening. If a person having the family history of genetic disorders and want to know whether he is a carrier for such genes or not, he recommended for karyotyping. To find out the reason behind the stillborn baby death and also to find out the reason for the miscarriages of certain women, karyotyping is required. To diagnose certain cancers and blood disorders, karyotyping is needed. Not only diagnosis, even we can make a treatment plan for certain diseases using karyotyping. Sometimes person might be not having any family history of genetic disorders, but he may acquire it in his lifetime due to mutations. To identify these acquired abnormalities and how they could pass to next generation, he needs help from karyotyping. I think you understood the importance of karyotyping. Now, how the test is performed, we will see. The first step in karyotyping is sample collection. We can use any tissue which has nucleated cells. But based on the type of diagnosis, we can use either of any from the following. They are amniotic fluid or placenta. Usually these are used for prenatal screening for pregnant women. Bone marrow and blood. Specifically, lymphocytes are used for karyotyping. Here is the pictorial representation of the procedure. Now we will see the flowchart for the procedure. After collecting the sample, dividing cells were separated and they are grown on the culture media for 3 days. Then the cells were synchronized at metaphase. Because at metaphase, chromosomes are at their most condensed state and we can visualize the rod-shaped chromosomes which allow us to identify the changes very easily. To do this, cells were treated with colchicine, which prevents the formation of the spindle fibers, thereby the cell cannot enter into the anaphase. Next, cells were treated with a hypertonic solution, which makes the nuclei to swell out and allows the chromosomes to separate and scattered freely in the nucleoplasm. Next step, cells were dropped onto the slide at arm's length distance, which causes the nuclei to burst on, and all the chromosomes were scattered on the slide, which creates a metaphase spread. These slides were subjected to fixation, then treated with trypsin to digest all the proteins so that chromosomes can take more stain. Here, usually we are using Giemsa stain to color the chromosomes. After staining, these chromosomes of a single cell were selected, photographed, and enlarged printouts were taken by using a special microscope which is fitted with camera and computer. Individual chromosomes were cut out from the printout and they are arranged in a standard decreasing order of size by placing their centromeres on a horizontal line which creates a karyogram. Look at the pictures. Here we can see a metaphase spread. When chromosomes are arranged in a standard manner which creates a karyogram which look like this. Now at this moment we can understand the formal definition of karyotyping. Karyotyping is a procedure of photographic representation of actual stained chromosomes which are arranged in a decreasing order of size by placing their centromeres on a horizontal line. Now let us see a human karyotype, how it looks. Human cells have 46 chromosomes or 23 pairs. Out of 23 pairs, 22 pairs are similar in both males and females. They are called as autosomes. Remaining one pair is called as sex chromosomes as they are determining the sexuality of an individual. In males, it is X and Y. In females, it is X and X. Arrangement of chromosomes is not random in karyogram. It should be in a standard manner which allows us to identify the abnormalities easily. Denver classified chromosomes in a proper manner. Denver established this system of classification in 1960. Denver classification is mainly based on the length or size of each chromosome and also based on centromere index. What is centromere index? The ratio of the length of the shorter arm to the whole length of the chromosome is called as centromere index. For better understanding of Denver classification of chromosomes, we try to recollect types of chromosomes based on the position of centromeres. Based on the position of the centromere, chromosomes are classified into 4 types. Telocentric chromosomes. In telocentric chromosomes, the centromere is located at the end of the chromosome. The chromosomal tips are called as telomeres, and these chromosomes appear as eye-shaped structures in the metaphase stage of the cell cycle. This type of chromosomes has only one chromosomal arm. Telocentric chromosomes are very rare in occurrence. Acrocentric chromosomes. The centromere is positioned near to the end. This creates one very short arm and one very long arm. Acrocentric chromosomes appear as J-shaped structures in the metaphase stage. Next, submetacentric chromosomes. The centromere is located near to the center of the chromosome. Thus, these chromosomes will have 2 unequal arms. Usually, in metaphase, they look like a L shape. Metacentric chromosomes. The centromere is located exactly at the center of the chromosomes. This creates 2 equal arms of a chromosome. These metacentric chromosomes will appear as V-shaped structures in the metaphase stage of the cell division. According to Denver, chromosomes can be divided into 7 groups, A through G. Here is the table which gives the information about Denver grouping. Let us see the classification pictorially. Group A includes 3 pairs of chromosomes, which are large metacentric chromosomes. Group B contains 3 pairs of chromosomes. They are large submetacentric chromosomes, but they are smaller than the Group A chromosomes. Group C is the largest group among the all. It has 3 pairs of chromosomes and also X chromosomes are included in this. All are medium-sized submetacentric chromosomes. Group D has 3 pairs of medium-sized acrocentric chromosomes. Group E includes 3 pairs short, either metacentric or submetacentric chromosomes. Group F, 2 pairs of short metacentric chromosomes are included in this. Group G includes 2 pairs of small acrocentric chromosomes along with Y chromosome. Here I am providing 2 karyograms. Have a look. One with XY chromosomes is male karyogram and other with 2 X chromosomes is female karyogram. Here, one thing we need to observe that all the chromosome centromeres are arranged on a horizontal line. Once karyogram is prepared, that should be analyzed by a cytogenetic analyst to find out the chromosomal abnormalities. Let us try to analyze some of the karyograms. Students, have a look. By observing the sex chromosomes, we can say that they are karyograms of a male. We can see one extra chromosome at 21st chromosome. This abnormality is called as trisomy, which results into a Down syndrome. Chromosome complement for Down syndrome is 45XY or 45 plus XX. In this picture, we can visualize the individuals who are suffering from Down syndrome. It causes developmental and intellectual delays. Let us have another karyogram. In this karyogram, we can see one extra X chromosome, which results into a Klinefelter syndrome. Chromosome complement for this is 44 plus XXY, total 47 chromosomes. Individuals are physically males, but they show feminine body characteristics. In the above set examples, we saw changes in the chromosome number. Now, in this case, we can see chromosome is moved from one chromosome to another, which is called as translocation. In this case, translocation is taking place between chromosome 9 and 22, which produces Philadelphia chromosome. This translocation leads to a cancer called acute myelogenous leukemia. Here I am mentioning some of the reasons for abnormal karyograms. Chromosomal aberrations includes both numerical and structural changes. Numerical changes are the changes in the number of chromosomes. Normal individuals have 46 chromosomes. Any change in this number considered as numerical change. Non-disjunction is responsible for numerical changes of chromosomes. Non-disjunction. Non-disjunction is the failure of homologous chromosomes to separate properly during cell division. This results in the daughter cells with abnormal chromosome number. If these abnormal gametes involved in fertilization that lead to production of abnormal individuals. Structural changes. If we observe any change in the structure of the chromosomes, those are considered as a structural changes of the chromosome. They are 4 types. Deletion. A portion of one chromosome is lost during cell division. Due to this, chromosome is now missing certain genes. When this chromosome is passed on to offspring, the result is usually lethal due to missing genes. Karyotype. A specific deletion of a small portion of chromosome 5, which results a syndrome called Karyotype. These children have severe mental retardation, small head with unusual facial features, and a cry that sounds like a distressed cat. Duplication. Part of a chromosome is repeated, resulting a longer than normal chromosome. Fragile X. Fragile X, the most common form of mental retardation. The X chromosome of some people is unusually fragile at one tip, seen hanging by a thread under a microscope. Most people have 29 repeats at this end of the X chromosome. Next, translocation occurs when one part of a chromosome breaks off and attaches to another chromosome, resulting in one shorter and one longer chromosome. An abnormal chromosome called Philadelphia chromosome is associated with acute myelogenous leukemia. The Philadelphia chromosome forms when chromosome 9 and chromosome 22 breaks and exchange portions. This creates an abnormally small chromosome 22. This leads to a acute myelogenous leukemia cancer. Next, inversion. An inversion occurs when a single chromosome undergoes breakage and rearrangement within itself. The most common inversion seen in humans is on chromosome 9. This inversion is associated with growth retardation, infertility, recurrent pregnancy loss, and cancer. Applications. Karyotyping is used for prenatal screening. To find out the reason for the death of stillborn baby, we need karyotyping. And also used to identify carrier screening for genetic disorders. To diagnose and also to make a treatment plan for certain types of cancers and blood disorders, karyotyping is used. To check acquired chromosomal abnormality, it also used to represent the evolutionary relationships and also to solve taxonomic disputes. I hope you understood how to identify the abnormal karyograms. Let us have a self-assessment. Here I am providing a karyogram. Identify the abnormality. In this karyogram, we can observe trisomy at 18th chromosome, which results into a Edwards syndrome. Usual symptoms are low birth weight and abnormal head shape. Let us go to next assessment. Try to identify it. In this, we can see only one X chromosome. It results into a Turner syndrome. Chromosome complement is 44 plus X0. Total 45 chromosomes. Usually, individuals having 46 chromosomes. But the individuals with Turner syndrome have only 45 chromosomes instead of 46 chromosomes. Usual symptoms are delayed puberty, infertility, and heart defects. Let us go to next assessment. In this, we can see deletion in one of the fifth chromosome. Due to this, its size is decreased. It results into a Karyotype syndrome. Infants with this condition have a high-pitched cry which sounds like that of a cat. Let us summarize once again what we learned today. We learned what was karyotype and why we were using this to know the abnormalities of chromosomes. We also focused on the procedure for development of karyograms. We discussed about Denver classification of chromosomes. We tried to differentiate normal and abnormal human karyograms using different case studies. We briefed reasons for abnormal karyotypes. We did self-assessment to check our understanding and also had a glance of applications of karyotype. Karyotyping is a test used to identify the abnormalities of chromosomes, usually performed during the genetic counseling. We learn how to make a karyogram. Here we are using colchicine to stop the cell cycle at metaphase and using hypotonic solution to burst the nuclei. Giemsa stain is used and enlarged printouts have been taken using specialized microscope. To produce karyogram, individual chromosomes are arranged according to their decreasing order of size. For arrangement of chromosomes in karyogram, we followed Denver classification system. According to Denver, 23 pairs of human chromosomes are grouped into 7. They are A through G. Group A, large metacentric. Group B, large submetacentric. Group C, medium-sized submetacentric. Group D, medium-sized acrocentric. Group E, short meta or submetacentric. And Group F, short metacentric. And at the last, Group G has smaller acrocentric chromosomes. Then we have seen normal, male and female karyograms and also studied cases of Down syndrome, Klinefelter syndrome and acute myelogenous leukemia. And at the last, we learned some of the applications of karyotyping. People often feel guilty about genetic disorders. It is important to remember that these changes cannot be corrected, exist throughout the life and pass to next generation. Sometimes families are very much aware of the genetic disorders, but they choose to ignore the fact without realizing that the outcomes can be dangerous. Problems of genetic disorders is increasing due to lack of awareness regarding genetic disorders and tests which can screen them. Lifestyle changes can cut the risk of genetic diseases. Eat well for good nutrition. Manage your stress. Exercise. Avoid smoking and other exposure to toxic chemicals. If we have family history of genetic disorders, the best option to have a healthy baby in future is to get screened before conception. Having a good knowledge of screening tests, well planning at right age, healthy lifestyle can give flourishing, blooming and vigorous next generation. Don't leave it on destiny. Let us build the healthy generation. Thank you.